I am Cameron Bertuzzi. Welcome to Capturing Christianity. Today we're talking about Christianity and the origins of life. And I've got two special guests with me, Dr. William Lane Craig, who is directly below me on the screen, and then Dr. Jim Tour, who is diagonal from me. I think he's over there. And the purpose of the show today, Dr. Craig recently finished the section in his systematic theology on abiogenesis. Dr. Jim Tour, he is well known on YouTube and elsewhere for his work on abiogenesis and his thoughts as a scientist and a synthetic organic chemist, his thoughts on the current state of the science of biogenesis. And so what we're doing today is just fostering a dialogue between Dr. Craig, between Dr. Tour on this topic, and I'm going to let them do most of the talking today. Dr. Craig, would you mind giving us a quick summary of some of the topics that you discuss in your book on abiogenesis as we start the program today?
Yes, let me begin by saying what a tremendous privilege it is for me to participate in this conversation with Jim Tour, who is an eminent synthetic organic chemist and a specialist in our topic today. Indeed, one might wonder what I, as a philosophical theologian, could possibly have to contribute to a conversation like this, given Jim's expertise. And I think there's a twofold contribution that I'm able to make. First of all, I'm able to bring a theological perspective to the question of the origin of life. And that complements, I think, or supplements Jim's scientific perspective. Jim has endured an enormous amount of personal invective and abuse for his personal religious views. And what people don't seem to understand about his work in this area is that, in fact, as a scientist, Jim Tour is a methodological naturalist. That is to say, he doesn't include religion or God in his science. He has said explicitly that I never appeal to God to explain the origin of life. I don't even appeal to intelligent design. He said, as a scientist, I would never say that we will never be able to explain naturally the origin of life on this planet. Rather, he simply remains agnostic about how life originated. He says we're clueless about how life originated on this planet. And that is the appropriate conclusion for a methodological naturalist. And so what I can do is to bring some theological perspective to the question here, because I'm not a natural scientist, I'm a theologian. And so I come to the table with belief in the existence of God already in hand and want to ask, how can I, as a Christian theologian, best integrate Christian theology with the best deliverances of contemporary science? The second thing I think that I can contribute is that you will discover that in talking with professional scientists like Jim Tour, technical jargon is so second nature to them that it can sometimes be very difficult for them to explain these concepts in terms that laymen can understand. And as a layman myself, I hope that perhaps I can contribute to the communication of these highly technical ideas in ways that our listeners can understand today.
So let me actually start. Dr. Tour, I'm going to bring you in here. I want to ask you a question, and we may cut this out and put this in a little short clip. But has science figured out how life began?
No, not even close. Not even close.
Why is that?
Well, we still haven't come up with a prebiotic route to the basic four classes of chemicals that we need. We need four basic classes of compounds, the nucleotides, the carbohydrates, the amino acids, and the lipids. Nobody has ever used a prebiotic route to get at those compounds where they have been enantiopure or even high enantiomeric excess. Then you have to polymerize them, and it's very hard to polymerize them. We've never come up with a prebiotic route to polymerize them cleanly, where we are controlling the relative chemistries that are needed to couple these together. And then once you've done that, you've got to somehow get them integrated into a living system. So even if people have tried to pack these things into a vesicle, but they don't start running any more than you could take the parts of a car and throw it inside into the driver's seat and expect the engine parts to start working. They have to be integrated in together. So all of this together becomes a real problem. So far, we're not even close. What people have done and claimed synthetic cells is to take a working cell already and make a copy of its genome outside the cell, remove the initial genome and put the copy of the genome into that cell, and the cell started operating. But the cell was already in place, and that's what they called a synthetic cell. But the life was already there. They just put in a different computer control chip, if you will, figuratively, in inserting the facsimile, the copy of the genome. So that's what was called a synthetic cell, but that's not making life. They already started with an existing cellular life. We're so far from being able to do this.
Yeah, and shouldn't scientists shouldn't they be able to agree, at least in principle, with your answer here? Like you're not saying that therefore naturalism is false or anything like that. You're just saying that as of the current status of things, we don't have that answer, and that's okay. Like maybe it's fine to not have an answer. Like, can't we just admit that?
Yeah, I think it's fine to not have an answer and to say we are nowhere close because nobody knows how to put those parts together to get them function again. And when you haven't made the 4 classes of chemicals, and people will say, well, look, I read this paper. But then when you look at the details of the paper, the regiochemistry was all different. They never used enantiopure materials. The homochirality of the polymers was not there. And so none of those parts could ever work anyway. So when you box them in, when you say, okay, I'll give you all the parts. We'll take an existing cell. We will extract from it something that's come from a natural living system. We will extract from the DNA, the RNA, the peptides, the polypeptides, which are all your enzymes and all your proteins, and then the carbohydrates, the polysaccharides. We'll extract from that. And so then also embedded in that is the key information on how to put these things together, because without the informational code, you're lost. And so it's already got the informational code because it's already come from a living system. Even if I just gave you those parts, could you put a cell together? And when they're in that corner, they will agree they cannot. They do not know how to do that. So they'll hum and haw and they'll shuffle around, but then they'll say, no, we can't do that. So you would think that they would say, yes, we're very far from this, but they keep saying we're very close to this. And I don't know why they do that. And as a scientist, it's not that I'm trying to be, this grand explanation here, give it a grand explanation. I truly cannot say that we will never solve this thing in a materialistic fashion. How can I say that? Scientists cannot predict the future. How do I know? If you had asked a person in the year 1700, would we ever understand where this information in a cell is stored such that when 2 parents are tall, their child is tall? And they'd have said, no, we don't know how. They would not have been able to fathom it. And then in the 1950s, DNA was discovered, the structure of DNA and how this code works within the DNA. And so now we understand. So several hundred years, you can have phenomenal changes. And the rate at which human knowledge, information in human systems, the amount of information that humans gather doubles every 2 years. By that, I mean, what took us 5,000 years of human history to accumulate will be doubled in 2 years from now. That's how fast information is accumulating. So all I can say is one day we might know how, but we are nowhere close yet.
So Dr. Craig, are there any theological implications of that kind of answer to this question?
I think there are, but let me comment on something else that Jim has said. I want to underline for our listeners that Jim Tour is not some kind of a radical conservative in saying this. Rather, the opinion that he has expressed is the judgment of many mainstream origin of life researchers. And I just collected a couple of examples for our listeners today. For example, the first one comes from Pierre Luisi, who is a Swiss origin of life researcher. In his text, The Emergence of Life, an important text on this subject, he says, you can have all the low molecular weight prebiotic compounds in the world, but you will not start life without ordered macromolecules. Those are those molecules Jim was talking about, the polypeptides, the polysaccharides, the nucleic acids. He says it is one of the unanswered questions, and it is a question that is not taken seriously enough in the literature. The fact remains that there has been no way to obtain ordered sequences by prebiotic means. Another origin of life researcher, Harold Morovitz, in his book, The Origin and Nature of Life on Earth, says we currently have essentially no understanding of what laboratory conditions would reproduce the emergence of life, which is exactly what Jim Tour said. Finally, I just read this week James Shapiro's book, this monster tome, Evolution. He's at the University of Chicago, and he says, with regard to the origin of life, that our understanding of this is nil. Those are his words. So the position that Jim is articulating here is one that is frequently voiced in the literature on the origin of life, and is not some sort of radical right-wing position. The other thing I wanted to say quickly is that Jim did use a couple of words that are technical jargon that most of our listeners won't be familiar with. For example, he talked about enantiomeric purity or enantiomeric excess. Now, what in the world is an enantiomer? An enantiomer is like a right and a left-handed glove. It's a molecule or two molecules that are mirror images of each other, but they cannot be rotated in such a way that they coincide. You cannot convert a right-handed glove into a left-handed glove by rotating it in space. And one of the intriguing things about the origin of life is that, for example, in these amino acid chains that form proteins, they're all left-handed. All of them that are enantiomers are left-handed. All the enantiomers are left-handed. And for that to occur simply by chance is virtually impossible. So explaining how you have these enantiomers all lining up in the same way is one of the great unanswered questions. Now, Jim also mentioned something called vesicles. What are vesicles? Well, origin of life researchers have exhibited enormous ingenuity in trying to explain how the original first cell formed. Life doesn't begin until you have cellular life. And the present cells that exist today are breathtakingly complex, far too complex to have come together by chance. And so these researchers envision that there might have been proto cells. And these are sometimes referred to as vesicles. This would be like an oil droplet that forms automatically in water. If you drop oil into water, and especially if you agitate it, you get these little globular vesicles. And the idea is that one of these vesicles kind of engulfed these biomolecules and formed the first protocell, and somehow that got cellular evolution underway and going. And so again, what Jim is emphasizing is that doesn't even come close to explaining the origin of cellular life and the complex functions in a cell. In fact, a vesicle will not do the trick. One of the problems with vesicles is that their membrane surface or their surface is impermeable. And so there wouldn't be any way for the cell to get in nutrients and put out waste. So I hope that makes it a little more comprehensible to our listeners what Jim was talking about.
No, I thought that was fine. You're really impressing me, Bill. This is amazing what you just explained. You've come a long way.
I watch your videos and read your articles.
But I just want you to know, Bill, that when I listen to your debates, I don't understand what you're talking about. I mean, you got all these philosophical terms, and
Yeah.
I really have to stop and play it back again and again to make sure I got it. When I speak my jargon, I'm just getting you back.
Okay, fair enough. That's
All
really
right.
true. That everybody who specializes in a field begins to use terms like epistemology and metaphysics
Yes.
and things of that sort almost unconsciously, and it inhibits communication. But you asked me, Cameron,
Yeah.
whether or not there's a theological perspective on the origin of life. And one of the most basic, absolutely basic distinctions I think that the theologian needs to make is the distinction between physical life, which is what the scientist is exploring, and non-physical life. Theologically, there is such a thing as non-physical life. God, finite spirits, human souls are alive, and yet they're not physical entities. The most solemn oath in the Old Testament that God uses is, "As I live, says the Lord," and then he will make some utterance. So God is a living being, but he's not a physical being. And so fundamentally, we've got to distinguish between physical life and non-physical life. And theologically, the origin of all life is God. God is the origin of a non-physical life. He was there in the beginning. And then the opening chapter of Genesis describes how God created life on this planet. Originally, the earth was, in Hebrew, tohu wa bohu, an uninhabitable waste. And then over the next 4 days, it describes how God transforms the earth to be a habitable biosphere for humans to live in. So theologically speaking, I think we want to say that God is the source of both non-physical and physical life.
Dr. Tour, is there anything you'd like to say with respect to what Dr. Craig just said?
Well, something that a lot of people ask me, and I try not to answer it because it's hard for me to get into other people's heads. I have enough trouble figuring out why I do and say some of the things that I do and say, let alone other people. But they often ask me, why is it? Why is it that so many scientists are seeing what you see, but not saying it? And I think this is certainly true. The origin of life people themselves, some of them are really good chemists. I mean, really good chemists. And they see what I see, but they don't say that. They'll say something, they'll make up a once upon a time story and talk about how this could have done that, and perhaps this has done that, rather than saying all the many, many problems that there would be. And why is it that the community is not speaking up? I certainly can understand why people would see it and say nothing, because they don't want to be attacked like I have been attacked. But why is it that these people who work in the area are seeing it and saying something totally different? Why do you think that is, Bill?
Oh my goodness. I am not sure, but I want to underline what you just said. I saw the most remarkable statement by Albert Eschenmoser about the origin of life and the failure to provide plausible scenarios for the origin of life on this planet. And his answer was that it is not the goal of origin of life research to explain how life originated on Earth, nor even how life could have originated on Earth. And therefore, the failure to do that is not a failure of the project. Rather, he says the purpose of origin of life research is just to show that living things can be assembled out of inanimate organic matter. And when I read that, I thought, well, that's absurd. We know just from our own existence that that can be done, that animate living things can be made up of inanimate material. We know that without any research whatsoever. And so this was, I think, the ultimate kind of buck passing with regard to the failure of this project, is to say that it's not a failure because it doesn't really try to explain how life could have originated on this planet.
Well, then, I have a huge amount of respect for Albert Eschenmoser. I have
Yes.
read his work since I was a graduate student, but yes, Albert Eschenmoser tried to make ribose so that he could start to build RNA. He was unable to make ribose. All he could make was the 6-membered ring sugars rather than the 5-membered ring sugars. And so he started to try to make an analog of ribose and to say, well, this will just streamline what we're doing. So he was totally unable to push this thing forward. Now, mind you that Albert Eschenmoser is in his 90s now. If he is still alive, he's probably in his mid 90s, and he did this work. I'm citing work that he even did in the 1970s and early 80s. It's come further than where he was at this time, but he had a terrible time with it. But that's very interesting. I have heard it said that we're not trying to explain how it occurred. We just have to explain how it might have occurred. But now he's saying
even that.
He denies even that. So that's a total confession that even that cannot be explained. So he's gone even further to make it into an answer where he can claim success, as you put it, because we already know that we're here.
Yeah. Let me say one thing about, for our listeners' sake, what Jim said about the importance of Eschenmoser's failure to synthesize this ribose. Ribose is the sugar constituent of DNA and RNA. It's called deoxyribose nucleic acid. And so this is one of those building blocks of the building blocks of life. The nucleic acids, the DNA, the RNA, are tripartite macromolecules that have a nitrogenous base, a phosphate, and a sugar, which is ribose. And in Eschenmoser's case, not only could he not synthesize the DNA, the nucleic acid, of course not, he couldn't even make the ribose, which is just one of the building blocks of the building blocks, as Jim has emphasized. One of the interesting things about Jim's work that does, I think, stand out is that while I think most of the origin of life researchers I've read would agree that we have no understanding of how the heavy molecular weight macromolecules essential for life were formed, namely those nucleic acids, the polysaccharides, the polypeptides, and the lipids. Those are your fatty acids. Nevertheless, they would say that we do know how the building blocks of these building blocks came to be. Amino acids, for example, are common in nature. They're found on meteorites. Similarly with sugars, these can be found naturally. And what Jim, I think, has contributed to the discussion is a profound skepticism that we even know how to synthesize the building blocks to make the building blocks. And that is, I think, an important point that he's making.
Yeah, so I can elaborate on that. If you consider the amino acids, the experiment of Miller and Urey in the mid-1950s was really an amazing experiment. to take small molecules, put high voltages across it, and distilling them up and heating this solution. And he got out a number of amino acids. People have gone back and analyzed some of these mixtures, and of the 20 amino acids, he may have gotten 10 of them. But the problem is that he got many, many other compounds along with it. That's a problem. And none of the compounds that he made had an optical purity or an enantiomeric excess. So in other words, you had both handedness. You had the left-handed and the right-handed there. Now, there are two amino acids that can self-crystallize into a pure form such that you would just get the L amino acid. The other ones don't do that. They crystallize as pairs of both the right-handed and the left-handed. However, you can get some of those other amino acids that don't crystallize well to crystallize on the surface of those first two amino acids that can self-crystallize into pure form. But if you look at the yields of those, they are very, very small. It's less than 1% of them. So in other words, if you have a mixture that's 50% right-handed and 50% left-handed, and 1% of one of those hands, say the right-handed, crystallizes out, you still have the vast majority of the mixture is still racemic. You still have the vast majority of the right-handed and left-handed mixture. Plus, these are mixtures. That is a huge problem. We find on meteorites, you can sometimes get very sophisticated molecules. Lots of molecules come in on meteorites, and they're never in high purity. And so what happens, not only do you get the left-handed and the right-handed most of the time, sometimes you can get a little bit of excess of one over the other. Sometimes it's as high as what I've seen, say numbers of 70% enantiomeric excess, which is still not good enough for a biological system. But let's just say these are what you get. They come as mixtures of many compounds. Like when you made these amino acids, they're mixtures. And why is this a problem? It's a huge problem because you can't get the chemistries to go on mixtures. It's like taking 10 different paint colors and mixing them together. And so now you have a beaker where you've mixed together all these different paint colors. And to say, well, now I just want to somehow pull out the red dye that is in there. It's very hard to do that. Now we have instruments that can do that, but it's extremely hard. No way on a prebiotic Earth could these be fished out. Same problem with what comes in on meteorites. All the sugars that have come in on meteorites, the nucleotides that have come in on meteorites, they're all heavily mixed together. Nobody has ever reproduced those mixtures. So what comes in is very small amounts because most of what comes in is going to burn up. But even you could reproduce those mixtures in a laboratory. You could say, okay, it has these 400 compounds. So, okay, we'll make those or we'll buy those 400 compounds on Earth and we will mix them all together. Can I use that mixture for anything? And the answer is no, because one molecule conflicts with another. The other problem with the amino acids now hooking together, even if you had all 20 amino acids, you have to now somehow get them separated. If you want to leave them together, what happens is you can't cleanly hook them together because they don't just have two prongs, an A and a B, coupling to an A and coupling to an A and a B. They don't do that because you also have a C prong there too. So half of the amino acids have a C prong that end up getting in the way. They end up coupling in the main chain. So all bets are off. Nobody has solved that problem. That problem is exacerbated with sugars, which are also called carbohydrates, which are also called saccharides. So sugars, carbohydrates, saccharides, they're all the same thing. That problem is much harder. So if you have glucose, just the simple molecule glucose can hook together over 1 trillion with a T, trillion different ways if you have just 6 molecules of glucose. How do you get just one? You have to have had enzymes to begin with. There's no way to cleanly do this chemistry. So all of this, every step we're lost on. We don't know how to make the glucose. Nobody's ever made even the simple molecule glucose. Nobody's ever made it. Never been made in a prebiotic chemistry sense, using things that would be available on an early Earth. You can do it today. You can do it with modern analytical and synthetic techniques. However, none of that would have been available on an early Earth. So you have to say, what's available on an early Earth? What kinds of chemistries might have been available? Let's try to put these things together. Nowhere close. Eschenmoser, in his whole career, could not make ribose, which is one carbon less, which is an even easier molecule than glucose. And so it's a problem at every stage. Every stage it's a problem. Again, that doesn't mean that there's no way this could have been done on an early Earth. We just don't know how. And even if we had all the pieces, we wouldn't know how to assemble them into a cell, because there's so much alignment that is needed. Not just the covalent detachment where one molecule hooks to another, but the non-covalent interactions. These are two molecules wanting to come close in space to each other. There's an ordering to these in cells that is highly complex that we didn't even know about until, I don't know, 15 years ago or so. It's called the Leventhal 2.0 paradox. How do you get this ordering to go, the non-covalent ordering? The numbers are utterly astronomical as to the combinations that one could come up with, and only a few of those combinations are going to work. That's it. And the Leventhal 1.0 paradox was, how do you take a chain of amino acids that is now a polypeptide and have it fold into the right shape it has to be an enzyme? Because you need other enzymes are called foldamers, which will fold that enzyme that you want into the right structure, because there's too many variations on how these things may hook up with one another. And so all of these are big problems. So we're just kind of lost on how life might have formed from these basic chemicals. We're just lost on this thing. And to project as if we're close, I think, is disingenuous. And I don't think it's accurate to suggest this to society. But what happens is these suggestions have come so far that most people, there have been polls taken, most people think that scientists have made simple cells in the lab, like a bacterium, like these prokaryotic cells, that these simple cells. People think that scientists have made this, and about a third of the people think that scientists have made have made complex animals like frogs. And there's nowhere close. We haven't even made the simplest of cells. And people will say, well, the original cells were really, really simple. Well, bioengineers have already figured out how simple a cell could be to work. And so they've already spelled that out for us, and we're nowhere close. Of the about 15 components that you need, do you know how many have been made in a prebiotic sense in a laboratory? Of the 15 or so components, 0, none of them have been made. Not a single one. Not only because these are highly complex structures that of organization of many molecule types. We haven't even made the 4 basic classes to assemble into those higher order structures, nor have we made even the simple molecules that build up those chains. And so we're nowhere close to solving this problem. We got a lot of work to do to solve this.
Dr. Craig, I know you probably have some things that you'd like to say to help clarify some of the things that Dr. Tuer just said, but I wanted to get some clarification. Someone in the comments is asking, who are you talking about? Is Eschenmoser? What was his first name? Was it Albert or Isaac?
Albert Eschenmoser, one of the great origin of life researchers, who, as Jim said, worked on synthesizing ribose in the laboratory.
Excellent.
Let me underline the importance of enzymes that Jim mentioned. How does the cell manage to do all of these things that we can't do in the laboratory? How in the world do these simple cells pull this off? Well, one of the ways is that they have these enzymes, which are protein products that help to run the mechanisms of the cell. But here you confront a kind of vicious circularity, because the enzymes, the proteins, are themselves the products of the nucleic acids, the DNA. And yet, in order for the DNA to function, you've got to have the enzymes. So you've got a kind of chicken and the egg scenario here, where in nature, in the prebiotic situation, there are no enzymes because there are no living cells to produce them. And yet without the enzymes, you can't have functioning biomolecules like DNA and RNA. And so there's a kind of vicious circularity there.
So we did have a question that came in. Would you guys mind if we turn to one of these questions? We've got a chemical engineer in the live chat, and he's wondering. He's got a question here. He says, "I'm a lowly chemical engineer, so take my question with a grain of salt. But some of this sounds to me like irreducible complexity type stuff, which seems like an argument from GAPS." Either of you have any thoughts?
I think that's quite incorrect. I'll let Jim comment on the science, but the irreducible complexity argument is an argument that organisms and organs could not evolve by chance if they have to have all of their parts in place in order to function. And that is not the argument here at all. The argument here is that there is no known way that these macromolecules could be synthesized in a prebiotic environment. It doesn't have to do with the irreducible complexity of these macromolecules, but simply with the fact that there are no known means of these things being synthesized naturalistically.
Yeah, in the sense that irreducible complexity means that you have to have many components in the same place at the same time. So that's often the hard part in an evolutionary scenario. Even in evolution, you have to have many components all happening in the same place at the same time in an evolutionary scenario, in that you can't have one piece evolving independently. It's going to be dependent on many other pieces. In a way, that is the same thing that's happening here in an origin of life. If you want to have the building of enzymes, you've got to have all 20 amino acids available in the same place. How do you do that? When there's mixtures, they're just going to randomly hook together and give you this cross-linking problem where that third prong starts hooking in. So you need other enzymes to keep that from happening. But in order for those other enzymes to form, they had to undergo the same reactions. What kept that third prong from hooking in? Nobody knows. Now, if you're going to say that the proteins came together through the help of an RNA template, well, where did the RNA come from? Because you needed enzymes to stitch together the nucleic acids to make the RNA. And then you needed other enzymes to assist each one of those amino acids coming in. And then you needed to have a code, a certain arrangement of the nucleotides in that chain to give you the code that you wanted. And the numbers are too big to have happened by chance. So, for example, if you have a number of over 10 to the 50th, that is so improbable. So 1 over 10 to the 50th or 10 to the minus 50, that's so improbable that could not happen in our universe. That couldn't happen in our universe. There's not enough time in the 14 billion years. Even if you had 100 of our universes, there's not enough time if you're up in it 1 in 10 to the 50. So if you even just had a protein that was 200 long, so you have 20 to the power of 200. That is way, way bigger than 10 to the 50th. So you got 20 amino acids, 200 long units. That's 20 to the 200. That's way, way too much. You can't get in that space. You can't get the combinations that you need to give you what you need to do any sort of synthetic work. And among those, you're going to have many that do degradative work, that degrade things. So this is what we're talking about. So in a sense, I understand what you're talking about, that yeah, you need many components in the same place at the same moment in time. And remember, these are just molecules that would be in a pool somewhere or something. And how did they all get in the same pool? How did you get the sugars together with the amino acids? Because if you were trying to make those sugars in the presence of amino acids, you never would have gotten the sugars in the first place, because the amino acids have the same type of alcohol groups, many of them hanging off. And those amines would compete in the same types of reactions. So you'd have to keep them separated. And so you have to have all of these separated from one another when you're trying to make them, and then later all come back together. How do you envision this? So it is a real problem. And as far as the gaps, if you mean a God of the gaps type argument, no, we're not suggesting that because we don't know it, we don't know how it happened, that it must be God. I have never done that. I never suggested in that way. I just say that because we don't know how to do it, we just don't know how to do it, period. That's not to say that therefore God must have done it, nor is that to say that we'll never be able to do it. I've never used that type of argument. I'm just saying I don't know how to do it right now. And it's not only that I don't know how to do it, the target is getting further away. And why do I say that? It's not like, okay, well, we've learned so much in the last 20 years, we're a little bit closer to solving this. No, what I argue is we're further away from solving this. We're further than we were 20 years ago. We're further than we were 50 years ago. And the reason I say that is because 50 years ago, we didn't know about the interactome problem, this problem of the non-covalent alignment that's going to be needed. So because as soon as we learned that cells have that and depend on that, now our target has moved way, way out. It's moved much further away. So the target has moved further away. Not that the cell got more complex, but our understanding of the cellular complexity allowed us to see that, wow, we are nowhere close to this. Because, in the 1920s, they thought a cell was just a bunch of protoplasm, a bunch of stuff randomly wiggling around in there. But now we know a cell is a factory. It's highly ordered, highly orchestrated. You have so many enzymes. You have thousands of enzymes working, which are nature's little nanomachines to put this thing together. And so the complexity has become so vast that the target has moved further away. So we're actually not getting closer with time. We're getting further away with time, which generally means that we're very far away from solving a problem.
Dr. Tuer, you asked a question to Dr. Craig a little bit earlier about why does he think that scientists are not willing to admit that we're so far away from this? And I was thinking a little bit.
He never really answered my question. He started going in and clarifying scientific points. So I just want to point out that you're pointing it out, yes, that the philosopher skirted the question.
Well, no, I wasn't trying to point that out.
That's true. I did skirt the question because I'm not prepared to psychoanalyze other persons to talk about their motivations. I don't do that. I assess arguments. I don't try to psychoanalyze those who disagree with me. But my point was that Jim's opinion is not a minority radical opinion. It's a common opinion in origin of life research that we have no understanding of how life originated on this planet. And I think that's really important for our listeners to grasp. Now, Jim's last comments give me a nice opportunity to introduce the theological perspective that I am eager to talk about. And so let me say, and this is in reply to Kevin as well, like Jim, I am not offering an argument for God's existence based on the origin of life. Jim doesn't do that because he's a methodological naturalist. As a scientist, he doesn't do theology. The reason that I'm not doing it is because I'm not doing what's called natural theology. Natural theology is that branch of theology that it attempts to argue for God's existence from religiously neutral aspects of the universe in which we live. I'm doing systematic theology, which begins with the data of Scripture, and with that in hand, then comes to the deliverances of modern science and says, how can we, as Christian theologians, best integrate our theology with the data of contemporary science concerning origin of life? And with respect to that question, origin of life theorists tend to fall into two broad camps, what we could call necessitists and contingentists. Necessitists think that life is the inevitable and necessary product of the laws of physics and chemistry. Given a universe controlled by our physical and chemical laws, it is inevitable that somewhere life will originate. Contingentists, by contrast, say, no, the existence of life on Earth is a highly contingent event that depends upon a conspiracy of highly improbable conditions that might not have obtained on any other planet in the observable universe. So you've got necessitists and contingentists with regard to the origin of life. And it seems to me that the theologian can adopt either one of these perspectives. You could be what I would call a theological necessitist. Namely, you would hold that in the beginning, God decreed the laws of physics and chemistry that would control our universe, as well as setting the finely tuned values of those constants and quantities to make the universe life permitting. And therefore, given the laws of chemistry and physics that God has established, it is necessary that life will evolve. That would be one theological perspective. On the other hand, you could be a theological contingentist. And here I think there are two forms that of contingentism the theologian might advocate. One would be creationism. The theologian might say that these highly improbable events upon which the origin of life depends are so astronomically improbable that they cannot be reasonably faced. And that therefore God has miraculously intervened in the sequence of natural causes to bring about the origin of life. And probably the most plausible form of this view would be some kind of progressive creationism, where God intervenes periodically in the history of the earth to bring about, through supernatural input, events that would not, in all probability, have ever transpired had he not done so. That would be creationism. The other form of theological contingentism would be what I call supervisionism. And this would be the view that without miraculously intervening in the series of secondary causes, God has nevertheless providentially supervised the unfolding of the historical processes so that this highly, highly improbable event would in fact come to pass on Earth. And probably the most plausible version of theological supervisionism would be Molinism. That is to say, God, by his middle knowledge, knew what would come about were he to establish certain pre-existing conditions. Usually, middle knowledge in Molinism is applied to God's providence over the free acts of human beings, but it equally applies to the situations in nature. God knew that if these contingent circumstances were created, then these highly improbable events would ensue. And so it seems to me that the theologian could be a contingentist, either of the creationist variety or of the supervisionist variety. When you assess these 3 points of view, I think probably theological necessitism is highly implausible. There just don't seem to be those intermediate stages leading to the synthesis of the building blocks of the building blocks, or the building blocks of life, or the first cell, that would have to exist in order to say that this is the inevitable and necessary effect of the laws of physics and chemistry. So I think we ought to go with some form of theological contingentism, either creationism or supervisionism.
Well, I hope you guys don't mind if I move on to another question that was sent in, and then I do have one last question that's going to close us out. So this one comes from Nate. It's a super chat that was sent in. Thank you for the super chat. Can you ask Mr. Tour, Dr. Tour, on his debate with Professor Dave? Also, Cameron, are you going to change the name to Capturing Catholicism? No, we're not changing our name at all. But let's get Dr. Tour. Let's get your the details on your upcoming debate with Professor Dave.
Professor Dave, yeah. So it's May 19th at Rice University in Houston, Texas, 2023. So 1 month from today. It's be a Friday night, and it's going to be in Keck Hall, room 100. And this is all up on my YouTube channel, Dr. James Tour. So if you just go to YouTube, Dr. James Tour, you'll see the details there. And there's only 250 seats in this lecture hall, 265 seats. We were limited because Dave and I each are going to have a screen, a big screen to display things on our computer. And that was the only a lecture hall that had dual screens. And So it'll be in that, but it's going to also be live streamed. So if you wanted to come and you didn't get in, you could just sit there with your smartphone and watch it. It's easy to log into the Rice University guest system and get free Wi-Fi and listen on in. And so that's what we're planning. And it's not traditional debate rules. I've never been in a debate. I've never been on a debate team. It's what Dave wanted, and I agreed to do this, is that we'll each have a 5-minute opening statement, or a 10-minute opening statement. And then one of us will ask a 2-minute question, and the other will have 5 minutes to respond to that question. And during that 5-minute response, there can be dialogue between the two of us. And then it would oscillate back and forth. And then after about an hour and 15 minutes of that, we would open it up for questions from the audience. It won't be the live stream audience. It would be the questions from the audience in that room, where they could ask either one of us, and each of us would have a chance to do that. So that's what we're planning.
Dr. Tour, would you be able to save me a seat? Because we're both in Houston. I might as well come out and support you.
Yeah, I the doors are going to open at 6. So if you get there at 6, you'll have a seat.
Okay. Well, I anticipate it's probably, I may have to line up before that. I anticipate that this is going to be a pretty well attended event.
Yeah. Well, I'll do what I can do, Cameron. I'm sure that night I'm going to be pretty busy. It's actually a lot more to doing this than I thought. But we're going to get through it.
Yeah, we'll see how it goes. Okay, we did have a very substantial super chat that was just sent in, and this guy's been sort of criticizing the stream the entire time. I don't know what his background is. Paul Rimmer's his name. He says, "Hi, Jim. So true that we are very far from a solution. We don't know what a solution would even look like. Isn't that some progress? Do you dismiss all recent prebiotic systems chemistry, or do you think some small progress is being made?"
Well, Paul, good to talk with you again. You are a devoted fan. Wherever I am, you are, and yeah, even that you are saying you work in the area of origin of life, and when you say we are very far from a solution, that gives me that makes me feel like I've accomplished something. That there are people who are working in the area of origin of life and agreeing with me that we are very far from a solution. When you say, will you dismiss all recent prebiotic systems chemistry? I don't dismiss all of anything. I mean, I'm certainly not even aware of all systems chemistry. I think that some of the things that are being done are really highly improbable that these things are occurring this way. So, for example, the work of John Sutherland, and I respect John Sutherland's talent as an organic chemist very much. The molecules that he has made with one hand tied behind his back, meaning that he was restricting himself to prebiotic chemistry conditions, I think is not realistic in the sense that he does use relay synthesis, like many people use relay synthesis. He can't deal with some of these mixture problems, and he brings in, bubbling of dropping water onto calcium carbide to happen to generate acetylene nearby so that you can make these vinyl monomers. I mean, these and then the very, very careful adjustments of pH up and down and keeping something cold and then all of a sudden having to warm it up, that these are not even realistic. So to me, it seems hard not to dismiss those. And according to some in the community, they agree with me. Certainly Shapiro did. This idea of this very, very careful chemistry. But I'm not dismissing everything. No, I'm not doing that. But you even said we are very far, and you capitalized very. So you're agreeing with me. So I feel good, Paul, that you and I are agreeing now.
So, okay, one last thing that I'd like to do before we close out the show today. We're about at the hour mark, and that's about all the time that we're going to have today. But I noticed, it occurred to me during the stream that what I've got here, I've got two very important figures in terms of Christian evangelism, but the two of you have very different approaches to evangelism. So what I'd like to do is Dr. Tour, I'd like to get what is your approach to evangelism when you attempt to evangelize, and then I'd like to get Dr. Craig's response to that and kind of explanation of what he does, and kind of compare and contrast the two approaches.
Well, I preach the simple gospel. The presentation that I just tell people the message that I heard, how my sin was exposed to me. It was clear that I am a sinner, and that there is nothing that I could do to save myself from being a sinner, that Jesus gave his life on my behalf, and that he died to give his life on my behalf, and then he rose from the grave 3 days later, and I take them through the resurrection of Jesus Christ. And this is the way I preach the gospel. And if I go 1 week without leading somebody to the Lord, I feel like it was a wasted week. If I go 1 week without leading somebody to Jesus in this way. And everybody that I share with, I get them hooked up in a 13-week one-on-one Bible study with a friend of mine, and then I get them in a daily Bible reading program. And so this is what I try to do, and that's how I evangelize. I never discuss origin of life. I never discuss age of the universe. I never discuss evolution. That is nowhere close to what I do when I share the gospel.
How about you, Dr. Craig? What is your approach to evangelism?
Jim Tour is a fruitful and accomplished personal evangelist. I am jealous of you, Jim, for the effectiveness of your personal ministry in leading people to Christ. My approach is public evangelism. I want to try to reach as many university students as I can. And it became clear to me very early on that the most effective forum for evangelism on the university campus is public debate, where you will have an event that brings together a Christian and a non-Christian to discuss fundamental issues like, does God exist? Who was Jesus? What's the basis of moral values? And in this way, I try to present the gospel in the context of giving an intellectual defense of the Christian world and life view, so as to try to open the minds of seekers and unbelieving students to the possibility that the gospel might really be true.
Yeah, I wish I could say...
Cameron, could I just mention, It's because of people like William Lane Craig that I I can see so many people in conversation coming to the Lord, because it's people like Dr. Craig that have laid the groundwork. One man sows, another man reaps. And so many people have gone before me to lay this groundwork. And it's like I just walk in and I just reap what all these people have worked at for many years.
That's actually what I was about to ask you about. Well, thank you both for coming on to talk about this subject. There were some people at the very beginning of this saying that this is like two titans coming together on this one subject, and it was like almost like Christmas for them. So this has been a real pleasure. Thank you both for joining us for this discussion.
The pleasure's been mine entirely.